Hello, everyone, and thank you for joining today's UMB ICTR Enrichment Seminar. Today, we have two guest speakers, both who have joined the ICTR in the last few months to add two new ICTR resources, computational biology through the ICTR Biostatistics Core and data asset management through the ICTR Informatics Core. Before we get started, just a reminder that this session is recorded and will be posted to the ICTR website in our education section for your reference. We plan to reserve the last 10 to 15 minutes for questions and ask that you please post them in the chat. And finally, as you leave this session, you will be redirected to a short survey. We value your input for future presentations, so please take a moment and share your thoughts with us. And now I am so pleased to introduce our guest speakers, Dr. Weichen Zhu and Dr. Patrick McArdle. Dr. Weichen Zhu is Associate Professor in the Department of Medicine at the University of Maryland School of Medicine. A leading genetic epidemiologist, Dr. Zhu specializes in the study of complex human diseases through cutting-edge omics technologies, including genomics, epigenomics, transcriptomics, and proteomics. Whether you're launching a basic science project or a clinical study, Dr. Zhu offers expert guidance across all phases of research involving omics methodologies, such as strategic study design, identification of relevant public omics databases and resources, grant and protocol writing, sample collection and processing, data analysis, and manuscript writing. Dr. Zhu can also serve as a mentor for career development awardees. Dr. McArdle is an associate professor with a PhD in epidemiology and holds dual appointments in the Department of Medicine and Department of Epidemiology and Public Health. Dr. McArdle leads the Data Coordinating Center for the NINDS SIGN Consortium and the Amish Research Program, where he guides data governance, integration, and analysis efforts that advance large-scale genetics research. Dr. McArdle's work combines proven methodology with a practical understanding of how data can drive better outcomes in clinical and translational research. Now let's welcome our guest speakers, beginning with Dr. Zhu. Dr. Zhu.
Thank you so much for the kind introduction. Good afternoon, everyone. So I'm Weichen Zhu. Today, I will introduce the omic research I have been involved and the advice or any consultation service I could help with your research. So today, I'm mainly talking about using omic in our biomedical research. And I'm just joining the biostat core of Actor, and this is our colleague in this biostat core. So I'm new to this core. I'm very excited. I'm looking forward to learn from all my colleagues, and also I'm looking forward to interact with you. So just a reminder that our biostat core offer all faculty access to up to 25-hour free research-related biostatistical service. And you can access to the service by fill out the form on the Actor website. If you can go to the website, and then there is a link on the web page in the biostat core service. And if you click that will link to the form to the right. And after you're filling out the form, we will start the consultation with you and the process. So my research focus has been using omic-related method to study complex disease. I believe most of you probably are familiar with omics now because it's such a hot area in biomedical research. Omic-related kind of refer to the various disciplines in biology that study entire set of biological molecules in an organism. So there's a different omic from genomic, the bottom layer, and then the epigenomic, transcriptomic and proteomic, metabolomic. So by the central dogma of molecular biology, we know that the genetic information flow generally from the genomic to to the transcriptome, and then transcriptome to protein. So that's how we kind of thinking about different layer of the omic together, and the different layer of omic give us a full picture of what's happening within the organism. The major characteristic of omics, as we can see, it can provide us with a multi-layer biological data, measure DNA, RNA, protein, and metabolites for a comprehensive insight into biological system. And comparing to traditional one molecule, one time research, omic approach emphasize pattern, network, and signature over individual molecule, so that it really gives us a holistic overview of the complex system and the mechanism underlying complex disease. With omics, it will also help us generate a large amount of data matrix, and that amount of data allows to do a data-driven analysis research rather than a hypothesis-based one molecule, one time approach. As you can see from the characteristic of omic, the advantage for omic in modern biomedical research is that it provides a comprehensive molecular profiling and now allow us to simultaneously measure thousands of molecule features, and that give us a tremendous amount of biological insight. Again, it will enable a data-driven novel discovery without the prior assumption. So it's really helpful for us to generate a new hypothesis. And lastly, not the least, it enable precision medicine by tailor the clinical measurement prognosis to each individual by using the large amount of data and information available for each individual. And that's kind of impossible with one molecule, one-time approach. I just want to give you some example of the application of omic in biomedical field. In oncology study, omic has been used for tumor classification and identification actionable mutations. And it can also used to cross host pathogen interaction for infection disease research. And in neurological study, which is my research field, omic help explore the complex mechanism underlying the disease risk and progression. Also in pharmacogenomic, that's a branch of the omic as well. It helps us to study how an individual's genetic makeup can affect the drug response variation. So all these can contribute to a genetic or omic level understanding of the disease, complex disease, and for us to better measurement the disease. So I'd like to give you some kind of flavor of some major omics that we will encounter in our research. The first one I'm talking about is genomics. This probably is the one that you're familiar with. The first one is genetic association analysis. You probably see this plot before. This is called the Manhattan plot. The x-axis is the chromosome, the position on the chromosome, from chromosome 1 to chromosome 22 and sex chromosome. And the y-axis is the log p-value of the genetic association um, with the particular disease outcome or trait, because it kind of looks like the building in Manhattan. So that was called Manhattan plot. Um, and usually the large amount of genetic association can be identified with population study and a large amount of a sample size. As you can see, in this case, show here was a 2018 genetic association study of stroke, ischemic stroke, with 60,000 cases. We identified 32 loci. And then in 2022, with the GigaStroke International Consortium, we assembled 110,000 cases and then found more locus associated with ischemic stroke. So obviously, those type of analyses need a large amount of sample size. So I actually do not expect that each individual lab will be able to conduct a very powerful genome-wide association study. Usually, I believe most PI and the lab will participate in a international consortium to contribute samples and data for a meta-analysis for the genetic association analysis. However, I do want to point out several applications of the GWAS related product that we can be used in our daily kind of a biomedical research, even with a small sample size. The first one is the Mendelian randomization analysis. Mendelian randomization analysis is an epidemiology method that uses genetic risk variant for a specific risk factor as a surrogate variable to determine if the specific risk factor cause a particular health outcome. So basically, it uses the genetic risk variant as an instrumental variable or exposure to study the causal relationship between the exposure here and the outcome. So the advantage is that the genetic variable that was fixed during conception, they do not independent with the environmental or behavior confounder later in the life. So make them less prone to confounding factors. So by using genetic instrument, the direction of the causal direction can only flow from genetic to exposure to outcome. So generally, there will be no reverse causality happen from outcome cause genetic change. So what we can usually do is using the public genome-wide association result that I show in the previous slides, and identify the genetic risk variant as a surrogate marker for the exposure, and then to say whether that the genetic instrument was associated with the outcome of interest. And this is would be a very powerful kind of analysis. And usually we can do this with the public available database without cost a large amount of funding resource. So I kind of really encourage you to look at this method. And the next is a polygenic score analysis. By polygenic score analysis, it's kind of related to the Mendelian randomization analysis I mentioned in last slides. So in last slides, I mentioned that we can use the risk genetic variable to say whether that associated with a particular outcome. With polygenic score analysis, we summarize or count the total number of risk allele in an individual across many genetic variants. Here I show example of 10 genetic variant, and then summarize, count the risk allele, and summarize the score, generate a score for each subject from A to L, and you can see, and then we can assess whether the polygenic score is associated with a disease outcome. So polygenic score analysis is particularly suitable for the complex disease, which were caused by many kind of multifactorial factors, including genomic factor is also multifactorial. So with the polygenic score, it provides kind of a summarized signature of the genetic risk for each individual. And then we can assess whether the polygenic score for a particular exposure would be associated with a particular outcome. However, there's some caveat. So it's important to control for the diversity of the population or the variation of the population ancestry in the data set, so that we will not be confounded by biased by the population composition in our study samples. And I want to point out, we do not need to have individual level data to perform the polygenic score association. We can use the summary level data, the GWAS association data, to conduct the polygenic score analysis. So that make it very convenient if you want to explore some hypotheses. For example, examine a particular risk factor is associated with another outcome. Besides the Mendelian randomization usage for the polygenic score, we can use the polygenic score to examine the shared genetic etiology among different complex traits. We can also dissect the genetic contribution from various sources to the complex trait, and also can use the polygenic score to investigate the gene-environment interaction. The other usage for polygenic score is to predict the risk of a complex disease or classify patient into subgroup to optimize measurement and treatment. Next, I want to talk a little bit about the transcriptome. I think by now, many researchers are familiar with transcriptome. So nowadays, previously, transcriptome was studied by using microarray assay, and now it's dominated by using RNA sequencing method. Usually, the workflow is to collect sample, purify the RNA, and then we process the sample in the DNA sequencing core. In the sequencing core, they will generate the sequence read, and then we align that to the reference genome, get the count or reading count for each genetic feature. For example, each individual transcript or each gene, combining different transcript isoform into one gene count. And then we run statistical analysis to compare the expression level of the gene or transcript between different conditions. And along with that, usually we'll do pathway analysis afterward, and also gene-gene interaction analysis, or protein-protein interaction analysis, network analysis. Just one kind of example using the transcriptome in stroke. There's one difficulty for a early on differentiation between two like different etiology of the ischemic stroke. One is the ischemic stroke due to cardiac embolism, where the thrombus formed inside the heart, and then it fall off and then block the brain vessel. Another etiology is the large vessel atherosclerosis that formed inside the brain vessel, and then block the blood supply to the brain. Clinically, the presentation can be very similar between these two different etiology. So it's hard to tell, and even some after uh, discharge and a long time kind of workout, there's 1/3 of the ischemic stroke without determined etiology. So that make the prevention of the recurrence difficult, because we couldn't tailor the prevention method to the etiology. So one use of the transcriptome is that we draw the peripheral blood from the patient, and then to see whether the transcriptome of the blood could differentiate the two etiology. And the answer actually is yes. So the first column is the non-stroke control sample, and then the next three are the cardioembolic stroke sample, and then the last three sample are from atherosclerotic stroke samples. So on the y-axis, it represents many genes. Each line represents one gene, and the gene expression level was colored by the heat map. So the higher the red has higher expression, and the green has lower expression. Um, so we can see the cardioembolic stroke, they have a high expression for this group of gene comparing to atherosclerotic stroke, but the low expression um, kind of lower expression for this group of gene comparing to atherosclerotic stroke. The transcriptome also give us opportunity to look into the mechanism of the systematic response to ischemic stroke. So when we look at the signature of each particular cell type within the blood, we found in cardioembolic stroke in the left panel, the neutrophil type of cell are dominated in the expression pattern, while the monocyte and the platelet, they have dominated in the transcriptome profile for the atherosclerotic stroke. And then usually with the transcriptome data, we will be able to look into pathway involved in each condition. Here we found that for cardioembolic stroke, the blood show a kind of a systematic immune response to pathogen-like stimuli. And to the right are the pathway analysis result for the atherosclerotic stroke. The coagulation-related pathway dominate the gene expression signature for this particular etiology. And that makes sense because the thrombus formation in the vessel does involve the coagulation process. However, even the coagulation process was involved in cardioembolic stroke, but it looks like the system treated the thrombus, the fallen thrombus, as a outside intruder, kind of like a pathogen, based on the analysis. So that makes that very interesting to see the system respond differently to different thrombus source. And the future kind of downstream laboratory research can follow up to see whether we can block the inflammation response to prevent the recurrence or increase the efficiency of the treatment. With the transcriptome data, we can also analyze the gene or protein-protein interaction network analysis with many molecules at the same time. So this particular example show a gene expression changed in the cerebellum of the brain after hypoxia environment stimuli. We found that many of the regulated gene actually they are commonly regulated by a transcription factor HNF4A. HN4A actually was found expressed in cerebellum based on the Atlas data available public. So that make the compelling that maybe what we see in the transcriptome are regulated by the upstream unobserved HN4A activities. So a natural follow-up for this transcriptome study is to study the HNF4A role in the hypoxia response in the brain. So another example is mesonomics. So this is related to epigenomic regulation of the transcription without involving change the genetic makeup. So epigenomic usually refer to the either the DNA methylation on the DNA sequence, or the histone modification for those protein wrapping around the DNA sequence. So both modification will change the configuration of the chromosome, and thus regulate the accessibility of the DNA sequence to transcription factor machinery, and thus either increase or decrease the gene expression level. So epigenomic usually refer to including DNA methylation and histone modification, RNA interference, and imprinting is part of the epigenomic regulation as well. Again, mostly the DNA methylation happening in the cytosine, guanine, CpG nucleotide enriched area. And that enriched area usually are in the promoter region of the genome. With DNA, the methylated nucleotide, it usually block the accessibility of a transcription factor to the DNA sequence. CpG methylation happening when the DNA methyltransferase transfer the methyl group to the cytosine nucleotide. Here is a kind of a real data from the DNA methylation array result. So it's kind of an overview of the methylation beta value. That's a methylated percentage for a particular loci. So this plot showed the beta value, the methylation percentage on the x-axis, and the y-axis is the density. So we can say the low methylation loci and the high methylation loci. And with this type of data, because on the array, they have a different chemistry for different loci, normalization is necessary to bring the data together in a consistent way. So the first panel is the raw data, show the distribution. And the last panel, actually the other different normalization method resulting the distribution change. And you can see the last one show a lined very well for the two type of different probes. I want to point this out because one different aspect of the omic analysis is to normalize the data before we can do statistical analysis. So the data pre-processing step is very critical. And similar to transcriptome data, we can use the heat map to show the differential methylation for different condition. Here, the x-axis are individual methylation locus across the genome, and then the y-axis are individual samples. The top samples are the pratic muscle from stroke patient, and this group of sample are from the non-pratic side of the same subject. And again, the color show the methylation values. You can easily see the different methylation pattern for different samples. And we can follow the analysis with pathway analysis. In this example, we found the methylation change in the pratic muscle actually is enriched with a gene that has a functional role in muscle-related function or neurological disease-related function. So that make the finding very biological, reasonable. So this is just kind of give you some flavor of what kind of omic research can be used in our research, and what kind of omic related consultation service we can provide. In our core, so I mainly in charge of the consultation for omic related approach. No matter what's your particular research area, we have the common analysis working flow and study design consideration across different research project. So that's why I can give you kind of advice to allow you to expedite your research using omic approach. Some challenge is that the omic data is a high dimension data. They often have more mirrors than samples, and there's a risk of overfitting and false positive finding in omic-related research. So we need to pay attention to the multiple hypothesis testing correction, particularly. Also, because the thousands, millions of features were observed, the data was complex. It make the data interpretation harder than traditional one molecule, one time research. So we need a particular a omic specific method to interpret the result and visualize the result. As we can see, use the heat map to show the different signature of the condition. We can, using the gene interaction network analysis to visualize the kind of interaction network of on the different condition, and also pathway analysis to see which signaling pathway was more important for a particular condition. Also, in omic research, because large amount data, any technical variability can be amplified, and resulting error in the finding. So we need to pay attention particularly to those batch effect and technical difference. And that's why we require a special normalization and batch correction for omic related data analysis. So if you could engage us early on in your project, then we can give you kind of advice from the beginning, from the study design up to data analysis and data interpretation, publication. And just to give you an example, what our service are, we can help on study design of omic-related research, and identify actually the relevant public omic database for you to use. Like the example I mentioned for Mendelian randomization and polygenic score association analysis. We can also do power calculation if you want to write a proposal or manuscript. We can also help develop a grant and a proposal and help you plan for your sample collection, biobanking, and processing workflow. Um, yeah, that's all from me. Um,
Thank you, Dr. Zhu, for that presentation. We're just running a little short on time, and we'd like to go ahead and move over to Dr. Patrick McArdle's presentation. Thank you again. And we'll save questions at the very last. And if anyone does not have their questions answered during this presentation, please feel free to reach out to us, and we'll connect you with either Dr. McArdle or Dr. Zhu.
Thank you.
Okay, ready to go, Rhonda?
Yes, please. Thank you.
Okay. Thank you for the opportunity to present today on data asset management in the era of collaborative science. This is a new service that's being offered by the ICTR's Informatics Core. It helps teams replace ad hoc exports and spreadsheets that we all have been using for our research and move more towards a traceable data pipeline and consistent cohort definitions with reusable data assets. So the problem is that in this age of collaborative science, that we've kind of outgrown this single research lab performing their own research on their own data. Collaborative science usually means that your data is going to be reused by analysts, investigators, and collaborators who do not necessarily generate the data, as my colleague Dr. Xu just mentioned. So the era of emailing spreadsheets back and forth has kind of passed us by, and we need a more scalable and governed system to track data assets in our research. So, as many of you are aware, the operational systems are not usually analysis ready. For those who have tried to get data out of REDCap in instances where using repeat instances, you probably know what I'm talking about here, but the REDCap and other sorts of operational systems are designed to store data in a way that is most amenable to their application. So the various capture systems, every lab or vendor that you work with has their own data formatting. Omics and high throughput data has its own formatting. They're inconsistent formats across and technologies used across these various platforms. So it's time-consuming rework used to every time you want to use the data for a paper, for a grant application, for analysis or a report requirement. That this core services aim to help alleviate those pain points. So the Informatics Core is launching this new service that help guide research teams towards developing a research-focused data assets. The resulting output of the consultation hopefully will be analysis-ready data sets that are easily cohortable, which is, able to create data sets for individual research questions. And obviously, the annual reporting and other sort of funding requirements that we all have to deal with could be streamlined quite a bit. So the service is actually just as much about process and governance as it is about technology. And it's a paradigm shift from one-time extraction towards a reproducible, reliable system that supports scientific analysis over the long term and collaboration between groups. So, reproducibility in science today is a design requirement. It's no longer optional. So this means creating rerunnable pipelines so that the same inputs produce the same outputs, preserves a record of what was received before any transformation may begin, and documents source to target mappings and validation rules, and has transparent issue tracking instead of hidden manual fixes. So the idea of just getting a spreadsheet and noting a data error and correcting it in the spreadsheet so that it is never documented or no one knows what was corrected is long gone. So the research payoff is really when results need to be refreshed, defended, or handed to another analysis, the work can be rerun instead of reconstructed. So what we do is we start with the data in the source systems and extract that data into some sort of landing area that preserves the original data, standardize and harmonize through some sort of transformation processes, and loads into a warehouse of conformed dimensions and facts so that the warehouse now acts as the single source of truth for all future either analytical data sets or reporting that goes on. So the idea is that we now have consistency across all the various outputs from your research organization. So, any of the reliable findings coming from your research labs require stable definitions and controls for it to be consistent over time and to be considered trustworthy in the collaborative sense. So, reliable findings are not just statistically strong, but also operationally dependable. That is, numbers are stay aligned across progress reports, manuscripts, and proposals. Ambiguity is reduced through metadata and data dictionaries shared with collaborators, and access can expand safely as collaborations grow. So there's a documented lineage of data, shared definitions across each of the aspects of your research area, and controlled access to all these data assets is another aspect of this core service that we would be providing. So here's a kind of overview of the research data supply chain. We start with a variety of various data from source systems, operational data from vendors or instruments or REDCap or API calls or any of that. You land it in a staging area, preserving the original data for traceability so that there is a provenance that is established, standardize and harmonize across these various different data sources, and then load into a warehouse, which then can be used for analytical data sets or reporting, as well as an optional document layer that could be added into this that includes non-structured data, things like IRB protocols, consent forms, things like that, that are necessary for proper interpretation of the data and govern the use of the data assets. So the source layer is where the research data actually come from. It's usually study-specific operational data sets and case report forms. They could be vendor or laboratory extracts, instruments or assays or omics outputs, and they have a wide variation in both the identifiers, the units, the code system, and unfortunately the common formats that are used. So you can get data, stored in REDCap, um, CSV files, Excel files, databases, API calls, JSON, custom flat file formats. It gets to be quite a challenge to manage all those different assets and create a uniformed, consistent data asset to produce output, such as publications or grant applications and analyses. So the investigator shouldn't have to solve these cross-platform integrations every time one of these outputs needs to be generated. The idea would be to create one process, one stored procedure, and to be able to rerun it, for example, when new data is generated and stored in REDCap. You might have a certain research cadence that says, hey, I'm going to extract data from REDCap every month or every quarter or every week and report on various metrics, maybe during recruitment phase of the study. And then during analysis phase, you may slow that cadence down and pull data from lab vendors and laboratories at a certain cadence. And all of this should be reproducible and reliable. So the key to the extraction and landing zone is it's a reproducible checkpoint where if at the end of this process someone says, I'm not sure where this data came from, you're able to point back to the exact landing point and say this is where the data came from at what time and when it was extracted, and establish some sort of provenance. So you never let curation overwrite the original evidence of what was received. You always store original data, of which you may have multiple copies. So it supports source reconciliation and allows transparent comparison between raw inputs and curated data outputs. And this, of course, improves confidence during audits or troubleshooting or hands-off when you have maybe staff turnover or things like that. So the transformation step is the key to the whole process. This standardizes the data across the various platforms that are used in your research and aligns it and harmonizes it, and importantly, documents all of that for source to target mappings to implementation rules that validate the data to generation of new data. For example, as an overly simplistic example, you may measure height and weight on everybody and calculate BMI during this process. And that, although that's a rather simplistic example, it ensures that consistency is applied for whenever a new variable is generated. For the next example, BMI is always estimated in the same way. So in that process, you can search for data quality and error, and then create a transparent error event log, which allows you to turn discrepancies into trackable and fixable events before your analysis takes place. So you can record the severity and the effective records, the date that was detected, the resolution status, all of these things that says, hey, we're trying to remove the probability or the possibility of having two different versions of the data that disagree with each other. So it creates a reviewable history for data managers, analysts, and investigators. So data warehousing has been quite popular in this big data era that we live in now. And it's no longer just for business applications, but it's for science as well. So it optimizes repeated questions such as stratification or reporting requirements. It relies on something called conformed dimensions to improve consistency across studies and time, and it stabilizes definitions for dashboards, feasibility work, and analysis use. So, um, if you imagine a pipeline where you're extracting all the data from these source systems, landing it in a space, transforming it, and then loading it into this data warehouse in a consistent, reliable, conformed manner, then you could create or use a variety of tools on that single source system for reporting or for analysis. And it's kind of technology agnostic. Once it's in this data warehouse, you can throw R at it. You could use Power BI dashboards to visualize things. You could either push or pull data sets in using CSV or whatever file formats you may want. So the kind of central hub is this data warehouse. So, important things to avoid when developing this data warehouse that we can help you and advise you on is that the wrong approach is simply to take the data that's given to you and put it into a warehouse. That is essentially skipping the transformation step. So, that will preserve the operational quirks and complexities that's in the data. So, a lot of the value in this process is the transformation and making the end result ready for analysis. And it encourages duplicated cohort logic and inconsistent measures in that case. So if you're getting data on the same individuals from different platforms where you have omics data coming from this vendor and you have data coming from the EMR and maybe some survey data that was collected on the same individuals, you'll need some way to harmonize all of that, organize it around time and assay and outcomes, and make definitions that are reusable across studies and outputs. So, as I said, once the data warehouse is created, creating analytical data sets is fairly trivial. You can either push or pull, depending upon your preferences and your data governance policies. That means that if you are working with a collaborator, you could either push a data set to them, or if you have an internal data analyst who is comfortable with the data warehouse, they could pull that data out to perform analysis. And it makes publishable data sets and views built on a shared cohort logic. And there's less rework when a table or a figure or a manuscript or a grant must be updated, so that when you submit a publication and you get reviewer comments and you need to regenerate that figure, but just with this one little tweak, well, great. You already have the process. It's going to be consistent. You just have to hit the rerun button and make any modifications that you need. Also, if you are working with a outside biostatistics team, like the ICTR Core or somebody, it makes for a cleaner handoff where you have ownership and control over your own research data and can create an analyzable data set for a biostatistician who does not need to go through all of that downstream work that really requires in-depth knowledge of your study design and your research area and your things like that for your definitions in your cohorts and things so that you have control over that. You manage that, and the data analysts are doing what they do best, which is statistics and inference. So the reporting layer, this is just a common pain point we all deal with, creating annual reports and even tracking like recruitment. As recruitment is going on, you may want to uh, have weekly reports generated this if there's certain maybe multi-centered research that you want to track recruitment by research center. It's all possible. So if all of the data is loaded into a common data warehouse, it's we have services within ICTR that will help you develop dashboards, for example, to layer on top of that data warehouse so that you can get near real-time look into the data and to display it how you wish. So once it's in the data warehouse, your display layer is open, can be whatever technology you wish to use. So finally, governance is a big um, aspect of this work. Um, there should be, and this is more process than it is technology, but there should be trust boundaries that must be built into the data asset lifecycle as who gets access to which data. You might have controlled access to your data warehouse for your collaborators and your biostatisticians, and for the raw data, you might have the internal analysts who can get to see certain data or not. So it comes particularly challenging when PHI is used, especially if Epic data is being used. Obviously, we all know that the secure research environment is now spun up and ready for open for business. So that will add a layer of complexity to it. But regardless, there needs to be some sort of controlled access to your data assets and to really treat your data as that, as an asset of your organization, where you can drive value out of that, whether that value is consistent publications, uh, more collaboration across campus or across the world, um, obviously grant applications and things like that. So all of that should be thought about in a global type of manner within your research group, so that governance makes it sharing safe and understandable and sustainable. So the scientific payoff is faster time to results because teams can reuse a governed foundation instead of rebuilding data sets. Greater confidence in published numbers because the numbers are stable across time and location. Easier collaboration across investigators and analysts and external partners since there's one source truth that we are all agreed upon, and improved readiness for larger multi-study and funded programs. So the study design and the data collection are only the beginning. As I said, if you have ever put data into REDCap and attempt to get it out to do any research, you probably know this pain point well. So durable data assets are what like good studies become reliable science. And that's all I have, I believe.
Dr. McArdle, that was wonderful. Thank you so
That was a
much
little
for
quick,
that
but I got it
presentation.
in under the clock.
Oh, now I'm just looking here to see if there are any questions in the chat, and I don't see that anyone has added anything. So we can follow up with you later if you like, but if you could get something in before the stroke of 1 p.m., we will be happy to take your question. If not, then we look forward to hearing from you in the future. I did add how to access these resources, the links for Dr. McArdle's services. You'll find that on our Informatics Core webpage. And then for Weishan Zhu's services, that's part of our Biostats Core services. And both those links are in the chat. Okay. I think that we can safely just let everyone move on and maybe grab a bite to eat or have the rest of your time for the afternoon. Everyone enjoy your day. Again, thank you so much, Dr. Zhu and Dr. McArdle. Those are both really informative sessions, and maybe we'll have a follow-up in the future. Dr. Davis, do you have any comments?
Uh, Thank you, Wanda. I just wanted to thank our two presenters, two wonderful presentations, very practical, very informative, and delighted to welcome you to the ICTR.
Thank you.
Thank you
And
very much.
Larry happened to squeeze in one question, still at 1 o'clock. Larry wants to know if the data warehouse creates data dictionaries.
Hey, Larry, how are
Patrick.
you? So the data warehouse itself does not create the data dictionary, but the data warehouse is one consistent part, is the consistent data asset of which you could build a data dictionary upon, so that there's only one single data dictionary for the study. But yeah, we can create scripts and things like that to create a data dictionary off of a data warehouse.
Wonderful. Thank you so much, everyone. And Larry, if you have any other questions, we'll catch with you later on. Thank you, everyone. Enjoy the rest of your day.
Thank you.
Thank you.